billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the
The voltage difference between the intracellular space and the extracellular space when a neuron is in its non-activated state is the cell's resting potential, which is around -70 millivolts. The resting potential is the electrical charge the cell has when it is resting and not stimulated by any open ion channels. The electrical charge inside the cell is more negative than outside the cell, due to the high level of sodium ions outside. Inside the cell are more potassium ions, but also negatively charged proteins. The cell membrane is filled with ion channels that allow ions to enter the cell when stimulated by an axial potential. When the cell is in its resting state, the channels are closed but still leaky, and ions will move in and out of the cell. The membrane is more permeable to potassium ions than sodium ions, and therefore more potassium ions will leave the cell because of the concentration difference created by the low level of potassium outside the cell. This will cause potassium to diffuse out of the cell. The potassium ions are brought back inside by the sodium-potassium pump that throws out 3 sodium ions and takes in 2 potassium ions with the help of ATP. The voltage difference between the inside of the cell and the outside will allow
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
The action potential, represented here as a bright light, originates from the axon hillock on the cell body. Once initiated, the action potential travels unchanged all the way from the axon hillock to the axon terminals. Events responsible for this all-or-none propagation of the action potential occur along its membrane. Here, in a cross-section of the axon membrane, we see the movement of sodium ions, shown as yellow spheres, into the cell as the action potential travels down the axon. As the action potential moves along the axon, brief changes in voltage across the membrane cause sodium channels, which are embedded in the axon, to open. Positive charged sodium ions then move from the outside to the inside of the cell. The positive sodium ions move inside the cell because they are attracted by its negative charge. After the action potential passes, the sodium channels close.
The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be re-established. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly.
At a chemical synapse, a neurotransmitter molecule, such as acetylcholine, communicates a signal from one neuron, the presynaptic cell, to another, the postsynaptic cell. Neurotransmitter molecules are stored in vesicles in the presynaptic neuron. Some vesicles dock, via specific docking proteins, at the presynaptic terminal. An electric impulse, called an action potential, arrives at the synaptic terminal and docked vesicles fuse with the presynaptic membrane, dumping neurotransmitter into the synaptic cleft. Neurotransmitter binds receptors on the postsynaptic cell, which, depending on the specific receptor and cell type, open or close ion channels. Thus, the action potential passes to the new cell. The empty vesicles are recycled via endocytosis of clathrin-coated pits and take up more neurotransmitter to start the cycle again.
Let's look closely at the organization of a synapse. The synapse consists of a button-like ending at the tip of the branches at the end of the presynaptic axon. This presynaptic button is separated by a gap from the dendrite of the postsynaptic neuron. The synaptic membrane of the postsynaptic dendrite has numerous sodium channels that are associated with proteins called receptors. Receptors can detect special chemicals released by the presynaptic axon terminal. When an action potential enters the presynaptic axon terminal, the terminal releases a special chemical called a neurotransmitter. The message encoded in the frequency of the presynaptic action potentials is converted by the presynaptic axon into neurotransmitter chemicals that are released and carry the message across the synaptic gap to the postsynaptic dendrite. The neurotransmitter chemicals bind to the receptor proteins associated with the sodium channels of the postsynaptic membrane. Receptor binding of the neurotransmitter chemical stimulates the sodium channels to open and allows positive sodium ions to flow into the postsynaptic dendrite. Transfer of the sodium ions to the inside of the postsynaptic membrane results in the outside of the postsynaptic membrane becoming negatively charged in the region of the open sodium channels. The negative depolarization of the postsynaptic area spreads over the sensitive postsynaptic region and generates new action potentials that are carried away by the postsynaptic axon. To stop the depolarization of the postsynaptic region by the neurotransmitter chemicals, proteins called enzymes are present that destroy the neurotransmitter chemicals. When the neurotransmitter chemicals are eliminated, the postsynaptic sodium channels close, the sodium is transported back to the outside of the postsynaptic membrane by the reactivated sodium pumps, and the
كيفية انتقال السيال العصبي بين خليتين عصبيتين. عند وصول السيال العصبي إلى الانتفاخات أو الأزرار تدخل أيونات الكالسيوم من خلال مضخات الكالسيوم وتروح إلى حويصلات التشابك وتعمل على تفجيرها ويخرج منها إيه؟ اللي هو الأسيتايل كولين اللي هو عبارة عن إيه؟ مركب من جزيئين يخرج منها الأسيتايل كولين. الأسيتايل كولين هيعبر شق التشابك ويروح إلى مستقبلات النواقل الكيميائية اللي هي دي ويتصل به. أول ما يتصل بها بتفتح مضخة الصوديوم زي ما هنشوفها كده ويدخل منها أيونات الصوديوم. واحنا عارفين أيونات الصوديوم اللي هي ديت الحمرا ديت لما بتدخل أيونات الصوديوم بتعمل إزالة للاستقطاب، يعني تجعل الغشاء من الداخل موجب ومن الخارج سالب. بكده انتقل السيال العصبي من الغشاء ده اللي بنسميه الغشاء قبل تشابكي إلى الغشاء اللي بعد تشابكي، يعني ننتقل من خلية عصبية إلى خلية عصبية أخرى.
A chemical synapse is a special junction between two neurons that allows them to communicate without being physically connected. A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft. An action potential causes voltage-gated calcium ion channels to open in the presynaptic terminal. The influx of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis. This neuron is releasing acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
How drugs affect the dopaminergic synapses. Drugs work by blocking either dopamine receptor sites or dopamine synthesis, and also by preventing the breakdown of dopamine. Dopamine is synthesized from tyrosine, a basic amino acid, and is stored in synaptic vesicles. Vesicles migrate to the presynaptic membrane and release dopamine into the synaptic cleft. The receptor stimulation of the postsynaptic membrane sends the impulse to the next neuron. Amphetamines and other CNS stimulants, like cocaine, activate dopamine release and effectively block reuptake at the presynaptic membrane. Dopamine activity stops as it is resorbed into the presynaptic membrane. Dopamine is then broken down by monoamine oxidase, MAO. When MAO inhibitor drugs, MAOIs, such as phenelzine, Nardil, and tranylcypromine, Parnate, are used, the breakdown of dopamine is prevented.